Hello again, everyone. Thank you for considering using SCIP for your CNV interpretation needs. In two previous videos, I demonstrated how to set up and use both the SCIP backend processing pipeline and the front-end visualization module. In this video, I would like to walk you through the CNV interpretation process using SCIP through an example. This example is publicly available on GitHub, so you can follow along on your own computer while watching this video. We previously published this CNV in a 2020 paper. This CNV was found in individual 033. He has the charge of Fallot and pulmonary atresia. To obtain this example from GitHub, go to the GitHub SCIP webpage, and there is a file called SCIP example here. And we can click the download link. I already unzipped the file to the downloads folder, and these are the contents of this folder. In order to try this example on your own, you must have both R and RStudio installed on your computer. For more information on this, as well as on the interface config.txt file, please see my previous video on setting up and using the SCIP front-end visualization interface. If you are using macOS and downloaded this zip file to the downloads folder, you don't need to modify anything before trying the example. However, if you downloaded this file elsewhere, or if you are using a Windows computer, you might need to modify the interface config file, specifically the temp file directory and the root directory entries. After you modify the interface config.txt file, you can double-click and open the SCIP interface.r script, and RStudio would launch. Click the Run App button here to further launch the SCIP application. We don't recommend you to run SCIP in this pop-up browser provided by R. Instead, we recommend using Google Chrome. You just paste that address into Google Chrome, and the same interface will be opened here. In this example, we are going to interpret only this CNV. For now, we can skip these sections. These sections are to be filled out once we completed the entire interpretation process. Moving on to the next section. This section provides a summary to the analyst about the overall information on this variant, as well as the positive and negative information. I will get into details about this just in a minute. Before that, we can see that this CNV has been rated by SCIP as a passed CNV. It was given a priority score of 2. This CNV was found in sample HSC 033001, found on chromosome 9. This is a deletion with 138 kb in size. You can use these links to go to the SAM region in the relevant tools. For example, we can click the Nomad SV link, and it will take us to Nomad SV, the exact SAM region. Moving on to the positive and negative information section, it gives an analyst a heads up about evidences supporting, which is the positive information, or against, which is the negative information, regarding variant pathogenicity and or quality. For this variant, we can see that there are a few positive information here. First, this deletion overlaps with gene with a high PLI score, which indicates computationally predicted high probability of being haploinsufficient. It also let me know that this deletion overlaps genes associated with dominant disorders, for example, Adam-Oliver syndrome 5, as well as aortic valve disease 1. If we scroll further down, we can see that this is quality information. This CNV is supported by 22 anomalous reads pairs and 12 split reads. Based on this positive information section, I know that this CNV appears to be overlapping some quite important genes, and it is well supported by anomalous reads. And reassuringly, I don't see any negative information. A major challenge in the clinical interpretation of CNVs is that the false positive rate in CNV detection is higher than that of SNV. So an analyst must evaluate variant quality up front while interpreting a CNV, and that is built into the logic of SCIP. Moving on to the next section is the read depths and mapping quality assessment. We can click the update read depths and update mapping quality. These two plots will now display. The design of SCIP is to plot the clinical sample of interest with a reference sample so that we can make sure if we do see any changes in read depths, it is due to the existence of a CNV in our clinical sample as opposed to a technical artifact. Here, the reference sample is NA12878, and you have the option to view sample only. Or, of course, you can view NA12878 only. So let's go back to the default view. The region highlighted by this light green shade is the CNV, and we can adjust the bin size here. The bin size is automatically determined by SCIP based on the size of the CNV to ensure speedy loading of the SCIP interface. However, you do have the option to change it. For example, I can change the bin size from 1 KB to 250 base pair, so you have more granular read depth information. I'm changing it back to 1 KB. The entire SCIP interface is interactive. You can say that I want to look at the left breakpoint, and you can select the region and double-click to zoom in. And in this case, you can say that I want to look at very granular read depth information, and you do have that opportunity to do so. Let's go back to 1 KB and go back to the entire CNV. Looking at this CNV, it does look like a true positive because we see a drop in read depths at both the left end and the right end of the predicted CNV breakpoint. And the same pattern was not observed in NA12878. Based on this section, and in addition to satisfactory mapping quality, I determined that this CNV based on read depth does appear to be a true positive. Then I can move on to the next section. So next section focusing on anomalous reads, and you can click the Load Anomalous Reads button to load this plot. CNVs are typically supported by anomalous reads. In this plot, it displays various types of anomalous reads. We can see that there are quite a few anomalous reads spanning the predicted CNV interval. Red color here indicates that these reads are read pairs facing towards each other, but the insert size is much larger than expected. The purple ones here are split reads, meaning that it is one single sequencing read, but part of it was mapped to the left end of the CNV, while another part of the read mapped to the right end of the CNV. Both of these types of anomalous reads can be observed for deletions. This is quite supportive that this CNV being a true positive. Of course, we can choose to zoom into the breakpoint and to make sure that the breakpoint call is accurate. And in this case, it is, because we see that all the split reads break exactly at the breakpoint. I can further zoom in. We can see that all the split reads break exactly at the predicted breakpoint. This is supportive that this breakpoint calling is accurate. Let's double click to zoom out. You also have the option to view particular types of anomalous reads. For example, if you want to only look at split reads, you do have that option. You can also customize the definition of insert site outlier. By default, this is set at 99.5, but of course you can make it more stringent. Set it to 99.9, and you can see there are fewer noise. Another part of this section that I did not mention is that all the information displayed in this figure is also displayed in the table format down here, so you can easily look up the detailed information about these reads. Let's try look at split reads only, and you can see that the last 5 digits of the read name are displayed alongside the reads, so we can search this 70592 here, and we can look at the details of this read. So we can see this is the entire read name. The left side of this read ended at this point. The right side started at this point. The gap between the left and the right start is 138 KB, which is consistent with the size of our CNV, and mapping quality is satisfactory at 60. the read type is split read. From this plot and the read depth plot that we just went through before, I determined that this is a CNV of satisfactory quality, and therefore we can move on to continue the assessment of clinical relevance of this CNV. Moving on to the next section, this section displays the external and internal variant databases. What we want to make sure here is that this CNV we observed is not a population variant. There are three panels within this plot. The first panel is the NOMAD SV panel. It displays this information of variants from NOMAD. The second panel is the ClinVar panel. It displays this variants reported in ClinVar, color-coded by pathogenicity. And the third panel is variants detected in our internal cohort. As you may recall from the previous video that we did not specify a cohort CNV file, so as expected, we're not seeing any variant in our internal cohort. And this is not important for this particular case because it is a singleton sequence. We do not have the parent's information. Let's start assessment from the NOMAD SV section. We do see a few deletions in this region, but their frequency appears to be quite low. We can use this NOMAD SV allele frequency to see if any of them are common. We can set the frequency to, say, 1%. And now we are immediately seeing just two of them. Both are very small CNVs, have a frequency above 1%. We are confident that the CNV we are seeing is not a population variant. Of course, we can also try slightly more stringent thresholds like 0.1%, and we are seeing also not a lot of CNVs. Let's just leave these four here, and I will get back to this. Of course, you can also filter by ClinVar consequence. You can say that I want to see pathogenic or likely pathogenic variants only, or you can say I want to see benign or likely benign variants only. In this case, we don't really see a lot of benign or likely benign variants. So this adds to our confidence that this does not appear to be a commonly encountered benign variant. You can also filter by ClinVar variant size. You can say that since our CNV is quite small, I only want to see CNVs with size less than 500 kb. From the assessment of this section, I'm confident that this CNV is not a population CNV commonly encountered. And all the information shown in this plot are also displayed in the table format down here. For example, we are seeing 4 NOMAD CNVs, and if I want to see more information about this CNV with ID del 9106735, I can go to here and I can click this and it will immediately take me to NOMAD. You can see that although it has a frequency of 0.005, it was only found in one NOMAD individual. It is not particularly concerning. Same thing for ClinVar. This table displays a wealth of information. For each ClinVar variant, you have the chromosome start and end and variant type information, the interpretation, the reported condition, allelic origin, and the gene it affects. I found this genes column particularly helpful because we can easily compare the gene content of our CNV with CNVs reported in ClinVar. And one-click links to ClinVar are also provided. If we want to see this CNV, we can click this link, and it will take us to the corresponding ClinVar page. Moving on to the final section here, the genomic neighborhood section. After assessing the previous sections, I concluded that firstly, this variant is of satisfactory quality, and second, it is not a population variant. We can use this genomic neighborhood section to assess the clinical relevance of this CNV. Click the Plot Genomic Neighborhood button would open this plot. We can see that firstly, triplet sensitivity is not applicable because this variant is a deletion, so we're not seeing anything in the triplet sensitivity map. This variant also does not overlap with any ClinGen curated haploinsufficient regions. However, NOMAD did predict 2 genes in this region, the SEC16A and Notch1 being haploinsufficient. We can see that this dark blue color corresponds to POI greater than 0.9 or LOF less than 0.35. So we know that they are bioinformatically predicted haploinsufficient genes in our CNV. This plot is fully interactive, so you can zoom in. For example, here, you can see these two exons belong to notch 1. We are quite familiar with notch 1 because multiple nonsense notch 1 variants have been previously reported in patients with non-syndromic tetralogy of Fallot. Let me zoom out. We can see that the entire notch 1 gene was deleted. Because our CNV is a whole gene deletion of notch 1, we do not need to review the PEX panel. However, if we are dealing with a partial gene CNV, we can use the PEX panel to assess the biological relevance of the affected exons. In other words, are they highly expressed? Similar to the previous sections, we also provide tables for all the information in the genomic neighborhood plot. Here we have notch 1, SEC16A, and C9orf163. We have 3 genes in this CNV. These 2 genes are located on the reverse strand. This one is on the forward strand. This is a full gene deletion for notch 1, a partial gene deletion for SEC16A, and a full gene deletion for C9orf163. For notch 1, we do see that it has a high POI score of 1 and very low LOF score of 0.09, pointing this being possibly haploinsufficient. We can see that it is highly expressed in our tissue of interest, which is fetal heart. And it has 2 GO terms possibly associated with heart. This gene is associated with Adam-Oliver syndrome 5 and aortic valve disease 1 in OMIM. These links are also directly clickable. You can click this and it will take you to the OMIM Adam-Oliver syndrome 5 page. And I also provided the GenCC curations. If you click this, it will take you to the GenCC page. You can see that it is curated definitive for Adam-Oliver syndrome 5, strong for aortic valve disease 1, moderate for connective tissue disorder, and supportive for bicuspid aortic valve. We can also use these links here to obtain more information about this gene. For example, I can click the clinical synopsis link and it will take me to the clinical synopsis page of notch 1. I can also click the allelic variant page and we can see that 1 base pair deletion, 85 kb deletion, and this nonsense variant and this 2 base pair deletion have all been reported as pathogenic. So we are more confident that haploinsufficiency is the mechanism for notch 1 related disorders. We can also use these links, for example, take me to GTEx, take me to Nomad gene page, this is GTEx, take me to ClinVar gene cards to further explore this gene. We can also use search terms to investigate further this gene with our disease of interest. I did not change the search term here, so it is not really suitable for congenital heart disease. However, if we say that we are studying a neurodevelopmental disorder case, we can click this ASD and it will take me to the Google search page, allowing me to search notch 1 and autism spectrum disorder. So if I'm not familiar with this gene, it allows me to search for all possible link between this gene and my phenotype of interest in Google. Another helpful feature of SCIP is the additional gene information pop-up window. This is a table in the genomic neighborhood section. For each gene, this pop-up window can be toggled by clicking the first, second, or the third column in the table. So let's click here. In this pop-up window, all the biologically relevant transcripts, as well as the canonical and main select transcripts, will be displayed. For each transcript, the exons that fully or partially overlap with the CNV are shown, as well as the percent of coding sequence affected by the CNV. For notch 1, we know that it is fully contained within this deletion. Therefore, we are seeing all exons from exon 1 to 34 out of 34 total exons fully overlap with the CNV, which corresponds to 100% of the coding sequence. For SEC 16A, this deletion only partially overlaps it. For the main transcript, we can see that exon 1 to 20 out of 29 or 28 are fully contained within this deletion, corresponding to roughly 87% of the coding sequence. This pop-up window is particularly helpful for partial gene or intragenic CNVs because we can easily assess how many exons and what percentage of the coding sequence is affected by the CNV, as well as for intragenic CNVs, whether it will result in an in-frame or out-of-frame change. The ClinGen dosage map tab here is not applicable for our case because there's no curated haploinsufficiency regions overlapping our CNV. However, if there is any, it will display it here and it will provide a link to ClinGen. Taken together, for this CNV, I determined that, let's go back to the top, that this CNV is of satisfactory quality based on read depth, is of satisfactory quality based on anomalous reads, it is not any common population variant, it is not a variant that's reported as benign in ClinVar. Instead, quite a few pathogenic or likely pathogenic ClinVar submissions overlaps or entirely contained within the CNV. And finally, it affects a gene Notch1, which we know that its haploinsufficiency would lead to congenital heart disease. For this reason, I determined that this CNV is pathogenic. So I can scroll up to the top. I can say that I determined the variant quality to be passed. I determined that this is a potentially reportable CNV, and I can enter my reasoning here. I can say that this is a deletion that fully contains the Notch1 gene. Notch1 haploinsufficiency is known to be associated with congenital heart disease. Variant quality was confirmed to be satisfactory. Therefore, this is a potentially reportable pathogenic CNV. And I can click the Save button here, and my interpretations here, including variant quality determination and notes, will be saved. I'm going to stop my video here. Just a brief recap. In this video, through this deletion containing Notch1, I showed you how SCIP can facilitate CNV interpretation by including all information in a unified and integrated interface. In the SCIP paper, we showed that it performed superiorly than both our previously used internal workflow as well as publicly available tools. Thank you very much for your attention.